My name is Mark Hannibal. I'm one of the team members for the Immuno-Hematology program, working in pediatric genetics. Our final session today is our selected best abstract when our committee got together and we're reviewing abstracts for inclusion as talks and as posters. We chose Gregory Baker's talk, submitted abstract for the best abstract submission. And Gregory is in a lab that came all the way from UCLA and transplanted himself here about 3 years ago, and I'll let him tell us about his talk and introduce it. Thank you.
Okay. Thank you, everybody. It's an honor. Thank you so much. Thank you, Mark, for the introduction, and thank Ashley Shaver for putting the whole thing together. It's been a really nice day so far. Again, my name is Greg Baker. One more. My name is Greg Baker, and I work in the laboratory of Dr. Pedro Lowenstein and Maria Castro. The primary appointment for our laboratory is in the Department of Neurosurgery. And today I'm going to be sharing with you the data from my dissertation project, which has elucidated what we believe to be a novel mechanism by which malignant brain cancers suppress the innate immune response. So when I first joined the lab, when we were at UCLA, the focus of my dissertation project was to elucidate the character of malignant glioma growth and invasion. And through these analyses, what we found very early on was the fact that malignant glioma cells tend to use the outside surface of brain microvessels as a pathway for tumor invasion. And we found that this was common not only in experimental models, but also in clinical biopsies of human brain cancers. So what I'm showing you here in this micrograph is a mouse glioma cell line called GL26 that's been engrafted into the brain of a transgenic mouse whose blood vessels express a fluorescent protein called GFP. And as you can see, each of these green cells are mouse brain tumor cells that bind to the outside surface and invade radially throughout the brain. So this was something that we were really interested in understanding the molecular basis behind. So for this, we analyzed cDNA microarray analysis from mouse GL26 cells that were grown in vivo. And through this analysis, we found a gene called galectin-1, which is a beta-galactoside binding lectin, which is to say it's a carbohydrate binding protein. We found that this was highly upregulated in GL26 gliomas and also in other perivascularly invading gliomas. So we became interested in studying the role of this gene in perivascular invasion. So we tested the expression of this gene at the protein level through immunohistochemistry, here shown in purple. And through the literature, we found evidence that suggested that galectin-1, or GAL1 for short, can regulate cell adhesion. And it also correlates with the degree of malignancy in brain tumor patients in the clinic. So we became excited to test whether or not we could suppress the expression of this gene, and if by doing so, we could block the perivascular invasion phenotype. So this is what we did. We used the mouse GL26 cell line, which is a prototypical perivascularly invading cell line, and we transgenically modified the cells to express a fluorescent protein called M-citrine so that we could see these cells using fluorescence-based microscopy. So we used shRNA-mediated gene knockdown to suppress the level of galectin-1 shown here through Western blot. And what we found was we were able to suppress gene expression by 60% in whole cell lysate. So this is total galectin-1 protein, suppressed by 60% compared to 2 control cell lines. And we asked a very simple question up front. We said, can we put these cells into the brain of a mouse and see if we could block the perivascular phenotype? So we did a very simple experiment to begin with. And when we did this, we found tantalizing evidence that this may be the case. These are 3 control cell lines, all showcasing the perivascular growth phenotype. And specifically within the GAL1 deficient cells, we see the failure of the majority of these cells to exit the needle tract in which they were initially implanted after 48 hours in the mouse brain. So this got us excited and was a first step forward towards identifying a role for galectin-1 in perivascular invasion. We moved forward by performing Kaplanmeier survival analysis in RAG1 knockout mice to see if we could find if galectin-1 could impart a survival benefit in mice that were implanted with these tumor cells. And what we found really surprised us. Shown at the top here is the Kaplanmeier curve. And what you can see is the Gal-1 deficient glioma cells failed to form lethal brain tumors even after 100 days post-implantation into the RAG1 knockout model. However, both control cell lines caused lethal uniform lethality within 20 days post-engraftment. So this really was startling to us. So we sectioned the brains of these mice and analyzed using low-power epifluorescence microscopy the size and the morphology of the resultant brain tumors in both control groups. And what you can see is large, massive tumor growth, shown in green, in each one of the mouse that are represented by the points on the Kaplan-Meier curve. However, after the 100-day time period of waiting for the mice to eventually succumb to tumor burden, we actually had to electively euthanize these mice and analyze the brain tissue to see if we could find evidence of tumor cells. And in fact, we needed to use high-power confocal microscopy even to see remnants of where the tumor had actually appeared to be involuted. The tumors never grew out. So this was a really strong phenotype. This may have been an initial indicator to us that maybe we weren't blocking perivascular invasion. This is something else going on here. So we moved on, and what we did next was do a detailed time course analysis, where we implant the Gal-1 deficient cells into the brains again of RAG1 knockout mice, and then ask the question, what is the morphology of the tumor over time, over the first 9 days? And this analysis revealed that beginning 3 days post-implantation, the M-citrine positive Gal-1 deficient cells become cleaved caspase 3 positive. Cleaved caspase 3 is a apoptosis marker, so these cells are undergoing apoptotic specific cell death that culminates after 9 days in complete tumor lysis. So these cells have actually been eradicated from the mouse model, and it seems like now we can be sure that we're not dealing with perivascular invasion blockade. It seems like we're dealing with a cell death phenotype. So we had to turn to the model that we were using. We were using RAG1 knockout mice initially because they lack adaptive immunity. Because these tumor cells express M-citrine fluorescent protein, which can be recognized as a neoantigen in wild-type mice who have adaptive immunity. We were restricted to using these mice to perform the Kaplan-Meier survival analysis I showed you earlier. But now we had to call into question the fact that these mice also have innate immunity. What is the role of the innate immune response on that phenotype of cell death that we saw? So we came up with a clever way of testing this question. So we incorporated the use of another immunocompromised mouse model called NOD-SCID IL-2 receptor gamma null, or NSG for short. Now these are the most immunocompromised mice on the market, so to speak, and they lack both innate immunity and adaptive immunity. So we posed the hypothesis. We said, if we were to assume that the innate immune response was responsible for the rejection of Gal-1 deficient glioma in the RAG1 knockout model, we would predict that if we put it into NSG mice, we should rescue the growth phenotype, and that's exactly what happened. Shown here. These are Gal-1 deficient gliomas engrafted into NSG mice, and we restore the lethal tumor growth phenotype. So we said it's the innate immune response. It must be the innate immune response. Let's move forward. We said we need to next ask the question, what cell types might be involved in this rejection? For this, we look to the natural killer cell. Natural killer cells are the effector lymphocytes present in the innate immune response, and they're one cell type that have played well-known roles in something called immunosurveillance, where they recognize transformed tumor target cells and directly lyse them. So we said this is a good place to start. Let's see if we can suppress natural killer cells in the brains or in the body of a RAG1 knockout mouse and see if we can again rescue the phenotype. And this is what we did. To deplete natural killer cells in the RAG1 model, we used an anti-serum called anti-ACLOGM1, which depletes natural killer cells in RAG1 mice, and B6 mice in general. And what we found is that when we did this and engrafted Gal-1 deficient gliomas back into the brain of the RAG1 knockout mice, each mouse that received the anti-ACLO-NK immunodepleting drug restored full tumor growth phenotype again, whereas all the controls shown above all rejected the tumor as we predicted. Now, we've done this experiment, not just the 5 mice that we've seen here. This has been done at least 50 times at this point through different permutations of this experiment. So we can really take this to the bank. We think that natural killer cells are responsible for this rejection. So we've gone one step farther now with a new graduate student in our lab, Peter Chalkley, who's performed immunohistochemistry for both natural killer cells, CD49B, which you can see specifically infiltrate Gowin-deficient tumors, shown here in green, and the natural killer cells are these small lymphoid-looking cells shown in black, which are just overwhelming the tumor and causing the rejection of the tumor. Now, in contrast, we see none of that in the non-target or the control tumors, which are growing out. These are on the same scale. Scale bars are the same here. Now, we also looked at something called granzyme B. Granzyme B is an apoptosis-producing protein, which is expressed in not only T cells, but also natural killer cells. Now, I'll remind you, we used RAG1 knockout mice for this. RAG1s don't produce mature T cells, so we assume that the granzyme B production that we see in this immunohistochemistry is specific to natural killer cells. And the quantification is shown at the far right. So we've asked the question now whether or not does this system work in a host that is fully immunocompetent? Does this work in a mouse model that has both innate and adaptive immunity? So for this, we used the C57 Black 6 model. And what we first did was implant GL26 Gowin-deficient gliomas into the brain of fully immunocompetent mice, and then treated them with either a sham, with either the sham normal rabbit serum antibody or an NK immunodepleting antibody. And then we assessed both the tumor burden as well as the level of adaptive immunity raised against the tumor through an assay called ELISpot, which is shown here at the center. Now let me focus your attention to the right first. In each case that mice had natural killer cells, we see after 16 days no evidence of tumor burden, which indicates that the mice through a natural killer cell function eradicated the tumor. Now when we look at our assay that asks the question, how many T cells have recognized that tumor, we see that very few, if any, or background signal is there, which indicates to us that the NKs do their job and get out before the T cells recognize the tumor. Conversely, in the mice that received ASGM1, or the NK immunodepletion antibody, you have brain tumor formation over the first 16 days that you never see in the RAG1 knockout model because we've depleted natural killer cells, and only after 16 days do we start to see evidence of the T cell immune response launch against the tumor to partly eradicate the tumor. And I'd just like to point out that it appears that this NK phenotype is the more robust phenotype. These tumors are completely eradicated in all mice, whereas you're getting a differential response in response to T cell mediated tumor eradication. So we've gone one step further and looked in vitro, and what we've been able to show is that not only do natural killer cells recognize and destroy Gowin-deficient tumors in vivo, the Gowin deficiency intrinsically sensitizes the glioma cell to NK-mediated tumor lysis. So for this, what we've done was we isolated RAG1 knockout natural killer cells and then stimulated them with 2 interleukins, interleukin 12 and 15. These things make the NK aggravated. It makes it activated and ready to kill. So what we've been able to show is that we can use 3.5 times less stimulation of our NK cells to cause 50% tumor lysis in a Gowin-deficient cell line compared to a control cell line. So it's showing not only are the tumors recognized by the cells, but they're also weak against the immune response. So to summarize what I've just told you, we've shown that natural killer cell immunosurveillance indeed can eliminate malignant glioma. This is what's new. This is the new component that we've shown. But it appears that galectin-1 blocks this process. We've further shown that when we knock down galectin-1, the cells are then recognized and destroyed by natural killer cells, and this all occurs prior to the adaptive immune response being initiated against the tumor. And furthermore, galectin-1 deficiency has been shown to sensitize glioma cells to NK-mediated tumor lysis. What are we asking now? We're asking the more difficult questions now is what we're asking. We're trying to find out whether or not, what is the causative agent that makes Gal-1 deficiency recruit NKs to the tumor and not in a galectin-1 expressing tumor. We're also asking what makes Gal-1 deficient glioma cells actually prone or sensitized to NK-mediated tumor lysis. And now we're beginning to start to think about translational implications of our experimental work. We're trying to understand whether or not we can come up with either small molecule inhibitors or gene therapeutic approaches to improve patient survival. And with that, I'd like to thank my supervisors, Dr. Pedro Lowenstein and Maria Castro, and the rest of my colleagues who work in the lab. Again, especially Vivek Yadav and Peter Chalkley, with whom I've worked very closely over the last several months since the two of them had joined the lab in regard to this project. And with that, I'd like to take any of your questions.
Beautiful talk.
Thank you.
Probably an obvious question, but since we know so much about the NK interaction with this target cell and the proteins that are involved, there isn't any chance that the knockdown of PD-1 is by terrible affecting the expression of any of these, exactly what it is.
So we've looked at MHC1, and we can't see any difference in MHC1. And we do want to look at a lot of the other co-stimulatory receptors, like the NKG2Ds and the ligands that would stimulate that. We haven't gone there yet, but this is something that we definitely have to look at.
You have more than one siRNA.
We have this. So we bought 5. These are commercially available, and we basically tested 5 of them and took the one that showed the best knockdown. I should say we also have clones. So this is the wild type cell line. You know, it's 60% reduction, but we can get it down to as much as 80, and we see the same phenotype. But I think what's neat about it is that you don't need complete suppression. So we're trying to ask the question now whether or not this is a cell autonomous effect, or if this is if a certain cell sitting next to a Gal-1 deficient cell can actually rescue the death of that other cell. So we're looking into it. Yeah. Yes.
Yeah, what is the purported mechanism by which Gal-1 inhibits? Is it attachment?
So galectin-1 is a carbohydrate binding protein. So in the 90s, in 1995, a UCLA group first showed that galectin-1 could bind to the cell surface of a T cell, of an activated T cell, through its interaction with N-glycans, on glycoproteins that are present on the receptors, activating receptors, and that this actually directly induced apoptosis of T cells. So the interesting novelty here is that people have tried to add, including ourselves, we've tried to add galectin-1 directly onto natural killer cells to see if it would affect their both lytic function and viability. And the answer is a resounding no. This has confirmed other people's work. So we don't know exactly what the mechanism is by which galectin-1 deregulates the innate immune response, but there's a whole wealth of information that suggests that it kills T cells directly through CD7, CD3, and a few others that I can't think of off the top of my head. Yes?
Have you tried serotherapy for galectin-1?
I'm sorry, how do you mean? Oh, antibodies for galectin-1. No, we haven't done that yet. So we have an experimental antibody that as you can see, we've used to probe for, Western blots and whatnot. We could try using that antibody. I know that other groups, there's a group in Buenos Aires by the name of Gabriel Rabinovich, who has blocking antibodies, who's published a nice paper in Cell recently using it. So maybe we could get a hang on that and we can try that. But I don't think there are any commercially available blocking antibodies. But there are commercially available small molecule inhibitors. So we can try those, but if we find that the result is negative and we don't get an effect, it doesn't mean that the drug is not working. It means we don't know enough about the mechanism by which we have a phenotype. So we're trying to understand better how the phenotype plays out mechanistically. Mm-hmm.
One more question.
Is there any direct or indirect evidence for these
Absolutely. Absolutely. Actually, we just have... I'm going to probably mess this all up. But I had, it's actually in the binder, but there's a number of Images at the, yeah. So let me show you just briefly here. So this is data from a database, a nationwide database on malignant gliomas. And what we can find through this database is that the clinical patients who overexpress galectin-1 in their brain tumors fare worse than the ones that underexpress it. And this is what the red bar shows you. The red curve is the overexpression, and the yellow bar is the intermediate expression. So there's the evidence for galectin-1 playing a role. But your question was not that. Your question was, can we find evidence for NK activating markers and things like this? This is NKG2D. So this is an NK receptor. And again, if we find that the NK receptor is upregulated, the patients do better, which is a good thing for us. Now, if we go on and we show, this is human MHC1. Again, if MHC1 is downregulated, they do worse. And it's not isoform inspecific. Look, all these isoforms, you see the same thing. So this is suggesting that maybe this innate immune response may be playing a role clinically.
Thank you.
Thank you.
Yeah,